Hey, what's up, biology students? Welcome to the virtual lecture on cellular respiration and fermentation, part 3. In this video, we're solely going to focus on fermentation. We've finished our discussion on cellular respiration in the last 2 videos. This video is also relatively shorter than the last 2, which I'm sure you'll be happy about. So fermentation is a metabolic process just like cellular respiration, and it is how some organisms, other than, let's say, animals and plants, so other organisms' means of, acquiring energy from the environment and converting that energy into a usable form that powers all their activities. This is another way of powering an organism. Now, it's not as efficient as cellular respiration. So we're going to break down the same molecule. We're going to break down glucose. But instead of generating upwards of 32 molecules of ATP per glucose metabolized, we're only going to gain 2 molecules of ATP. So that's what I mean by not as efficient. But that's okay, because these are different types of organisms that are undergoing fermentation. They're not as large and complex and multicellular as plants and animals. So the first question we want to ask, why does an organism undergo fermentation? What is the point? And the point is the same as cellular respiration. The point is to generate ATP. Remember, ATP powers all life forms, all living things. So that is still the goal, because remember, ATP is this convenient, usable form of energy that powers all cells, whether you're one cell or whether you're a trillion cells. Our next question is, what are the types of organisms that do fermentation? I'm reading this and I fixed it. It's a typo. It should read, what type of organisms do fermentation, not cellular respiration? And the answer is primarily single-celled organisms, many types of bacteria, also yeasts, which are funguses, and surprisingly, even animals like yourself can undergo fermentation in certain tissues for very short periods of time. But primarily we're talking about yeast and many different species of bacteria. And those are examples of single-celled organisms. So that's what I meant by, even though we're not getting 32 ATPs per glucose, we're only getting 2. That's okay, because the whole organism is only one cell, so 2 ATPs is just fine to power all of its activities. Now, before we move on with these kind of like essential questions, we're going to watch a fun video on the importance of fermentation in both bacteria and yeast for humans, on various food industries and various, I guess you could say, alcoholic beverage industries. So their importance on bread, cheese, wine, and beer. Okay? These foods and beverages wouldn't be possible without the fermentation being done by single-celled organisms like bacteria and yeast. So let's take a moment to watch that.
Where does bread get its fluffiness, Swiss cheese its holes, and what makes vinegar so sour? These foods may taste completely different, but all of these phenomena come from tiny organisms chowing down on sugar and belching up some culinary byproducts. Let's start with yeast. Yeast are single-celled fungi used to make bread, beer, and wine, among other products. Yeast break down carbohydrates like sugar to get energy and the molecules they need to function. They have 2 different ways to do this: the oxygen-dependent or aerobic pathway and the oxygen-independent anaerobic pathway, which is also called fermentation. When you bake bread, yeast can use both pathways, but they normally prefer to start with the anaerobic process of fermentation. In this process, ethanol is produced in addition to CO2. No, bread isn't alcoholic. Small amounts of alcohol that are secreted evaporate during baking. In the aerobic or oxygen-dependent pathway, the yeast consumes some of the sugar and produce carbon dioxide gas, or CO2, and water. In both processes, the CO2 accumulates and creates tiny bubbles. These bubbles get trapped by gluten and create a sponge-like structure that gives the bread its soft texture. Wine also relies on yeast, but a winemaking setup keeps the oxygen levels low so that yeast consume sugar using fermentation, the anaerobic pathway. The process often starts with wild yeasts already hanging out on the grapes. But to get consistent results, most winemakers also add carefully selected strains of yeast that can tolerate high levels of alcohol. The yeast consume the sugar in the grape juice, and as the sugar level drops, the alcohol level rises. This doesn't necessarily mean that sweeter wines have less alcohol. Different types of grapes start with different amounts of sugar, and sugar can also be added. What happens to the carbon dioxide? It just bubbles away through a vent. In carbonated alcoholic beverages like champagne and beer, sealed containers are used in primary or secondary fermentation to keep the carbon dioxide in the bottle. Wine also introduces us to our second type of food-producing microorganism, bacteria. A special strain of bacteria turns a tart compound in grape juice into softer tasting ones that are responsible for some of the flavors in red wines and Chardonnays. Another type of bacteria, called acetic acid bacteria, isn't so desirable in wine, but they have their function too. If there's oxygen around, these bacteria convert the ethanol in wine into, well, acetic acid. Let this process continue, and you'll eventually get vinegar.
Yeah.
Bacteria are the key for cheese, too. To make cheese, milk is inoculated with bacteria. The bacteria gobble up the lactose, a kind of sugar, and produce lactic acid along with many other chemicals. As the milk gets more and more acidic, its proteins start to aggregate and curdle. That's why spoiled milk is clumpy. Cheesemakers usually add an enzyme called rennet, naturally found inside of cows, goats, and some other mammals, to help this process along. Eventually, those little curdles turn into bigger curds, which are pressed to squeeze out the water and create a firm cheese. Different strains of bacteria make different kinds of cheese. For example, a species of bacteria that emits carbon dioxide is what gives Swiss cheese its characteristic holes. Some cheeses, brie and camembert, use another kind of microorganism too, mold. So your kitchen functions as a sort of biotechnology lab, manned by microorganisms that culture your cuisine. Yogurt, soy sauce, sour cream, sauerkraut, kefir, kimchi, kombucha, cheddar, challah, pita, and naan. But maybe not all at the same dinner.
So thank goodness for yeast and bacteria that undergo fermentation. That narrator also has a really nice voice. I need to practice to sound like that. I gotta turn on my YouTube voice. But anyways, we'll get to the human relevance of fermentation in the PowerPoint slides in a bit. But first, we have to talk more about the cellular or biochemical aspect of the process. So our next question is, why do some species undergo fermentation? Well, the answer is, at least for bacteria, which remember are prokaryotic organisms, they don't have organelles in their cells. So for instance, they don't have a mitochondria where the citric acid cycle or the electron transport chain can take place. So they just can't do it. Remember at the start of the first video on metabolism, We refer to cellular respiration as an aerobic process, meaning that it occurs in the presence of oxygen. Fermentation is the opposite. It does not occur in the presence of oxygen, it actually occurs in the absence of oxygen. Many of these organisms actually are killed if oxygen is around. Not all of them, like for instance, as you saw, yeast can go both ways. It can do cellular respiration if there's oxygen around, and then if it's deprived of oxygen, it can do fermentation. But fermentation itself is anaerobic. And in terms of the pathway of the fermentation process, it's quite simple compared to cellular respiration. It's basically just the first stage of cellular respiration. It's just glycolysis by itself with one extra step. So that's nice because we've already covered glycolysis, so it's somewhat of a review. And since we're not going to go past glycolysis, we can say that all of the other stages of cellular respiration, like stages 2, pyruvate oxidation, stage 3, the citric acid cycle, and then stages 4 and 5, they all shut down. Okay, so those don't occur. And since we're solely talking about glycolysis, this entire process is occurring in the cytoplasm. So nothing is happening in the mitochondria. So before I read this last paragraph here, it's easier for me to draw it out to show you what's going on. And we're going to draw out the effect that the absence of oxygen has on the ETC to show that causes a buildup of a certain molecule. So let's assume we're talking about yeast, which can undergo cellular respiration, or for instance, maybe the muscle cells of a human or an animal. So this is a picture that we drew out in the last video. It's the stage 4, it's the electron transport chain. And what we're going to do here is we're going to imagine that this cell is deprived of oxygen. So we're going to say, let's get rid of the oxygen. We want to understand, what effect does that have? And the effect that it has is that the ETC is a series of electron transfers, and oxygen is the final acceptor of electrons. And if there's no oxygen around, what happens is every preceding carrier of electrons is kind of stuck holding a pair of electrons with no one to give them to. So for instance, if you work your way back, the next carrier, cytochrome C, it has nowhere to donate its electrons, so it's stuck holding this electron. And then if you work your way back, the next carrier, ubiquinone, also cannot transfer its electron because, well, cytochrome C already has one. And if you work your way back all the way to NADH, same thing. Ubiquinone already has an electron, so NADH is also stuck holding on to electrons. So to summarize what occurs here is that you have a buildup of NADH, right? The electron shuttles, and they have nowhere to deliver their electrons to. So in essence, the entire electron transport chain shuts down, and you can no longer generate 28 molecules of ATP using chemiosmosis in stage 5. So all of this becomes obsolete, and we have a major problem because we're stuck with these NADHs and nowhere to give their electrons. And that's a problem because, remember, these molecules are shuttles. They go back and forth. So if you want to continue to do glycolysis and get 2 ATPs there, you have to free up these NADHs, turn them back into NAD+, so they can go back and pick up another pair of electrons. But you can't do that if they're stuck holding electrons. So our question here is, How do we solve that problem? Where can we dump these electrons so we can continue to use NADH to do glycolysis? And the answer is really fermentation. That's the name of that step. So we're going to draw that out. This is our second and last drawing of this entire video. All right, so we're going to start here at the top left. So remember, in fermentation, just like cellular respiration, the molecule that we're breaking down or metabolizing is also glucose. So let's label that. This is our glucose right here with its many pairs of electrons, right? It has a lot of energy to tap into. And remember, glycolysis is still pretty complex. It's like 10 steps. We're going to summarize that whole process, right? And what happens is we split glucose in half, generating 2 molecules of pyruvate. And in the process, we're going to produce 2 molecules of ATP in a very special way that doesn't require oxygen. So we're going to write that. We're going to get 2 molecules of ATP. We're going to get 2 molecules of pyruvate, which is, in a sense, the 2 halves of glucose. And if you remember back to video number 1, the third product of glycolysis is 2 molecules of NADH. And I kind of want to show you how that works. So just after glucose gets split, we start to oxidize the 2 halves of glucose, breaking bonds, stripping electrons, and removing energy. And we actually strip away 2 pairs of electrons. So imagine we do this twice. We break bonds and we strip away 2 pairs of electrons. So I'm going to show you the result of that. So I'm going to get rid of this, and I'm going to drag this over here. There we go. But remember, we have 2 of them times 2. All right, that's great. For this organism, we, met our overall goal, which was to generate ATP. We got our 2 ATPs, but now we're stuck with these 2 molecules of NADH. Now, you're never just breaking down 1 molecule of food at a time. You're breaking down like thousands and millions. So there's going to be a next glucose that's going to come in that you need to break down in the same manner. But in order to do that, you need NAD plus again in its form where it's not carrying a pair of electrons. So how do we free up our NAD plus? Basically, where can we put these electrons? We don't care about them because we're not doing any of this. So let's imagine that this was an organism undergoing cellular respiration. What it normally would do, if you remember from the last video, is your NADH would be shuttled into the mitochondria, and specifically, it's a little big, drawn to scale, but specifically it would donate its electrons to the ETC, and those electrons would be used to generate a massive amount of ATP, specifically 28 molecules of it. But again, we're not doing that because remember, we're in an environment right now where there is either very little or no oxygen. So let's indicate that. No oxygen. So the ETC shuts down. So there's no point in trying to use these electrons to do anything because the ETC is dysfunctional at this point. So all we want to do is just get rid of these 2 electrons that are sitting on top of NADH. The answer to our question is these electrons get donated actually to pyruvate. Because pyruvate, in a sense, in fermentation, is a waste product. Because we're not going to do pyruvate oxidation, we're not going to do the citric acid cycle. So if we're not going to use this, well, who cares if we just toss 2 electrons onto it? It's a waste product. And that's exactly what the yeast or the bacterial cell does. It takes these electrons and plops them right onto pyruvate. So let's see if I can do that neatly. Might be difficult. Well, I failed my first attempt. Okay, I'm just going to have you imagine that they get transferred. So we're taking these 2 electrons, and they're getting transferred to pyruvate. So just picture that in your mind. The result of that would be NAD plus again. And we can keep doing glycolysis over and over again, each time generating 2 ATPs. Now there's one other thing I have to say. What becomes of this pyruvate once electrons have been added to it? Does its name change? Does it become a different molecule? Is it relevant to humans? Yes, to all 3. It's not relevant to the yeast or the bacterial cell. Okay? So what happens is that pyruvate either becomes alcohol, aka ethanol, same alcohol in wine and beer, or it becomes lactic acid, depending on the specific type of fermentation that the organism undergoes. Those are the 2 different types. There's alcoholic fermentation and there's lactic acid fermentation. So I'm going to try to squeeze that in here. So once pyruvate has gained a pair of electrons, each pyruvate becomes either alcohol or lactic acid. And like I said, these are waste products from the perspective of the yeast or the bacterial cell. This does nothing for the organism. They're not doing fermentation to produce alcohol. They're doing fermentation to get energy, so they can interact with their environment, so they can build their own molecules and do everything that a cell needs to do. That's why they're doing this process. These are byproducts. These are analogous to carbon dioxide in cellular respiration. You know, it's useless to us as a human. We exhale it. It's a waste product. These are the waste products of fermentation. But it just so happens that, you know, humans have found many uses for them, as you saw in our previous video, for making different types of cheeses, for producing wine, beer, champagne, and also bread. And if you were doing alcoholic fermentation, where you're producing alcohol, ethanol, another byproduct of that is CO2. So again, these are 2 different types. You either produce alcohol and CO2 or just lactic acid. So that's fermentation. There's nothing else to it. It's basically, again, just glycolysis. You get 2 pyruvate, 2 ATP, 2 NADH, plus one additional step where we get rid of those electrons that were on the NADH, transfer them to pyruvate, generating these waste products so that we can continue to do glycolysis with new molecules of NAD+. So that's fermentation. We're going to go back to the PowerPoint slides. Now this last paragraph here should make more sense. I'm going to read it. It says, the absence of oxygen results in a buildup of NADH with nowhere to deliver their electrons, right? They can't deliver it to the ETC. Therefore, these NADH molecules, generated by glycolysis, instead transfer their electrons to pyruvate, so that future rounds of glycolysis can continue to produce 2 ATP per glucose that is metabolized. Simple, but efficient for a single-celled organism like a bacterium or a yeast. On the right side, we have a review question that reads, where would NADH normally deliver its electrons to during aerobic cellular respiration? I actually showed you that in my drawing. The answer was, well, if this organism was undergoing cellular respiration, which it's not, those electrons would be transferred to the ETC. So the ETC is the answer to that question. Next, we're going to take a brief, closer look at the two different forms of fermentation. The first being lactic acid fermentation. So that's when your pyruvate, upon gaining a pair of electrons, is converted into lactic acid. This is the type of fermentation that your muscle cells can do for brief periods of time when you're doing a really strenuous workout and oxygen is limiting, very low. But it's not something that the cells of your body can use as their primary means for generating ATP. It's kind of a desperate thing to do when you are, like I said, undergoing a strenuous workout. This specific form of fermentation is also the primary form of fermentation for many species of bacteria, also some types of funguses, and we use some of these species to help us make cheese and yogurt. For instance, in yogurt, it is the lactic acid that you're tasting that gives it that bitter, tart taste. So that's why I have a picture of Swiss cheese and a picture of Greek yogurt. The other form is called alcoholic fermentation. Very similar. NADH transfers its pair of electrons to pyruvate, but slightly different chemistry goes on in the reaction, and for whatever reason, a carbon is stripped away from the pyruvate and it exits as a molecule of CO2, leaving you with CO2 and, in a sense, lactic acid minus CO2, which we call alcohol. So in this form of fermentation, we produce ethanol, which is, again, alcohol as we know it, and carbon dioxide. Important for causing bread to rise, giving bread its sponginess, giving beer its carbonation, and also its alcohol. As you saw in the video, the same type of yeast pretty much are used in the making of bread and beer and wine, but your bread doesn't have alcohol because it evaporates during the baking process. And your wine doesn't have carbon dioxide, carbonation, because it is intentionally allowed to dissipate from, I don't know if they're called vats when you make wine, the apparatus that you're making your wine in, the barrel, I guess, I don't know. So, organisms that undergo alcoholic fermentation, the most important for humans would be yeast, but also many species of bacteria do this as well. And it's interesting to point out that yeast, which again are single-celled funguses, yet really complex organisms, because since they're funguses, they're eukaryotic, so they have many of the same organelles that our cells do, yet they're just one cell. But the interesting thing I want to point out is that they can do either fermentation or cellular respiration depending on the environment that they're in. So in a brewery, they're forcing the yeast to undergo fermentation. So they're purposely depriving them, suffocating the yeast, forcing them to undergo fermentation so that they produce CO2 and alcohol. But if there's oxygen around, those yeasts can do cellular respiration just like us. And our last few slides here are to round out and summarize this entire unit and to have a better picture of where this all fits into your entire metabolism. Because remember, metabolism in a biological sense is a very broad term. It pertains to all of the different types of chemical reactions that you undergo in your various cells. Thousands of different types. Here, in this unit, we're specifically focusing on the area of metabolism that relates to acquiring energy from food, whether you made it as a plant or whether you consumed it as an animal, releasing that energy and storing it as ATP so that we can move and pump and, what's the third one? Build bigger molecules, anything that requires energy. So, what else is there to metabolism? I want to point out that that's not it. Cellular respiration is not everything. So even though we focused on the breakdown of glucose, you can obviously use fats to generate ATP. You can use, sometimes in desperate situations, proteins, amino acids to generate ATP. But all of these other biomolecules, whether they be fats or proteins, they all funnel in to various stages of cellular respiration. So that's why it's convenient to focus on glucose, because it's kind of the cornerstone, it's the foundation of your metabolism. So let's see. We just said this. Please realize that glucose is not the only digested food molecule that we use to generate ATP. I would say that it is the preferred small molecule for all life on Earth to produce ATP, but it is not the only. We certainly use fatty acids. We certainly use the backbone of fats called glycerol. And we definitely use many amino acids which are derived from proteins that we ate. So just realize that. And I'm going to give you a few examples in the next 2 slides of how we metabolize fats. So the first step in breaking down a fat occurs in your digestive system. You have enzymes that use hydrolysis that basically chop off the 3 fatty acid tails from glycerol, so that you have 2 smaller components. You have fatty acid tails and glycerol. Then those really long fatty acid tails, which are just a bunch of carbon and hydrogen, they're chopped up, in a sense, using enzymes, into little pieces of 2 carbon molecules. So for instance, your average fatty acid tail is like 18 carbons long. So that means in this process, you would chop it up into 9 little 2 carbon chunks. You don't need to know this, but the name of that pathway is called beta oxidation. Main point is, when you break down a fatty acid, you get tons of these little 2 carbon chunks. You can funnel all of these little pieces of your fatty acids into the citric acid cycle, as you can see here, and they enter as acetyl. So instead of a derivative of pyruvate, you can skip that and it's like tossing like little logs into a fire or a furnace. And if you imagine that the mitochondria and the citric acid cycle is the furnace, you can toss in little logs of energy from here or from here. So yeah, that generates a tremendous amount of ATP. Also, the glycerol, which is 3 carbons, is funneled into glycolysis. Next slide, focusing on carbohydrates. Remember that in order to metabolize a carbohydrate, you first have to digest that polysaccharide into its constituent monosaccharides. And all those monosaccharides are converted into glucose, which starts at the very first step of cellular respiration, which you should be familiar with because we just covered it in the previous 2 videos. So basically, if you're eating table sugar, aka sucrose, lactose, which is milk sugar, fructose, which is found in fruits, doesn't matter, or glucose, you're going to metabolize it in the same way. It's all going to end up as a bunch of glucose. It's going to be broken down via cellular respiration, stages 1 through 5. And then my last little point here is that amino acids can be converted into a variety of molecules depending on the specific amino acid that you're dealing with. Remember, there are 20. And they can enter either the citric acid cycle or glycolysis or pyruvate oxidation. So you can use them and funnel them into pretty much every step of cellular respiration. And I think I said the last slide was my last slide. This is my last slide. It's also important to note that you can take molecules out of metabolism, out of cellular respiration, and you can use them for building if you wish to do so. So you can take pyruvate, you can take components of the citric acid cycle, and you could pull them out and use them to build your own fats, build your own carbohydrates, build your own proteins. So it's quite complex. Like I said at the start of video number 1, this really, truly is the study of nutrition. Okay? So nutrition, even though a lot of people like to say they know a lot about nutrition, is very complex. It's biochemistry, it's organic chemistry. And this is it, you know. How we metabolize our food and how we convert that into ATP, but also how we pull molecules out of the process and store them as fats or store them as glycogen. And that rounds out the unit. Thank you for listening.